So welcome everyone, and thank you for joining us today. My name is Thalia Eley, and I work as a research associate at the Wolfson Center for Young People's Mental Health. It is my pleasure to welcome you to today's lecture, which is part of our public lecture series, and I believe the fourth one for this year. So the lecture will be recorded and will be posted in the Wolfson Center YouTube page, where you will be able to see any other talks in this series that you may have missed. This session is scheduled for an hour, and questions and discussion are encouraged. So please post your questions in the Q&A box, which you should be able to see at the bottom of the Zoom screen. So our speaker today is Professor Thalia Eley, who is a professor of Developmental Behavioural Genetics at the Institute of Psychiatry, Psychology and Neuroscience at King's College in London. She directs the Emotional Development Intervention and Treatment Lab, and her work focuses on the interplay between genetic and environmental factors in the development and treatment of anxiety and depression. Her work is highly interdisciplinary, using both twin design and molecular genetic approaches, and drawing on cognitive, clinical, and developmental psychology. So for today's talk, it will focus on longitudinal explorations of developmental and treatment outcomes in anxiety and depression in adolescents and young adults. So over to you, Thalia.
Right, I'm just going to share my screen. Is that coming up okay?
Yeah.
Great. Just want to move this out of the way. And this. Okay, so good afternoon, everybody. And yeah, I'm going to present some work we've been doing using longitudinal data to explore the development and psychological treatment outcomes for anxiety and depression, primarily in adolescents and young adults, although some of the data relates to broader adult cohorts. So as you all know, sorry, I've just got this right over the top of my screen trying to work out where to put it. As you all know, the scale of anxiety and depression is pretty substantial. Around a third of individuals are going to experience one or other of these conditions during their lifetime. They begin young. The mean age of onset for anxiety disorders is 11 years, and for depression, it's around 18. So by the time children and adolescents have left secondary school, most of those who are going to experience anxiety or depression have already had their first experience of it. It has really significant impacts on learning outcomes and on education and on functioning generally, which is one of the reasons why it has become increasingly of interest in the past decade to try and understand how to prevent and treat these conditions better. And one of the arguments behind one of the major treatment efforts in England and in the Talking Therapy Service was a financial argument made by Lord Layard and David Clark, and this shows projections of costings in billions per year for 4 major psychiatric diagnosis categories, so depression, anxiety, schizophrenia, and bipolar disorder. And you can see that the dark blue section of each bar, which is treatment costs, is roughly similar. That's because although schizophrenia and bipolar are rarer, they are more expensive to treat, and it's more common to have inpatient treatment. But depression and anxiety have these huge on costs, so costs of loss of earnings, of benefit uptake, broader costs to the family and to the society, which mean that they result in a much greater cost to the economy as a whole. And it was this argument that was used to persuade the government to initiate what's now called the Talking Therapy Service. So I'm going to talk about different levels of risk in my presentation today. I'm focusing on what we think of as individual differences, so identifying how individuals differ from one another, thinking about that in terms of demographic characteristics, in terms of clinical profiles, but also in terms of genetics. And because the genetics part may be new to some of you, I know it isn't to some of you. I saw some familiar names in the attendee list when I looked just now. But for those who genetics isn't so familiar, I'll just do a quick whistle-stop tour through the methods and why we use them and what they tell us. So first of all, just some basic thinking about how genetic factors influence complex traits such as anxiety and depression. So genetic differences are called alleles, and so at any one locus on the chromosome, you could have two different alleles, an allele 1 and an allele 2. And if allele 2 is associated with a slightly higher score on a trait, then you might expect people with 2 copies of allele 2 to score a bit higher. And then if you had a second allele B, which could come as B1 or B2, and again version 2 is the increaser, then those with an A2, A2, B2, P2 genotype might be expected to have the highest score. And this combines in greater and greater detail as you have more genotypes that you're including, and eventually you end up with something that's basically a normal distribution. And so this is how we think of genetic risk for traits such as anxiety and depression. We believe it to be normally distributed in the population, and we expect that those with disorders are typically going to be at the higher end of this distribution, but not necessarily. That's just the overall picture that we expect. Because there's another important piece, which is, of course, the environment. And this is a useful infographic from Robert Plomin's Behavioural Genetics Primer as a way of explaining how we think about genes and the environment in the context of population variation. So if you think of an individual rectangle and you want to know its area, then its area is the product of the breadth and the height of that one rectangle. For this population of rectangles, if you want to know about differences in their areas, you need to know about differences in height because they don't vary in breadth. Whereas for this population, they're varying in breadth, but not in height. And the reality is that we expect a population of rectangles to vary in both breadth and height. And so we need to know about both to get the full picture. And this is how we think about genes and the environment, that we need to explore both in order to get a full picture of what we're looking at. So for the longest period of time of the work we've been doing, it's been the twin design that we've used, and then other designs have come in more recently. So just to quickly summarize the twin design, you're comparing resemblance in identical twin pairs with resemblance in non-identical twin pairs. And there's assumptions in the twin design that both types of twin pairs end up similar to one another because of shared experiences that make them similar. So, for example, living in the same family, going to the same school, living in the same socio-demographic kind of area. And this will lead to some resemblance that's of an equal size for identical twins and non-identical twins. Then in addition to that, there's resemblance because of sharing genetic influences. And this will be twice as great for identical twins who share all their genes as it will be for non-identical twins who only share half the genes that differ amongst humans. And so this difference between identical twin resemblance and non-identical twin resemblance is roughly half the heritability of the trait. And that's, in short, the basics of the twin design, and everything else builds from there. We typically model twin data using structural equation models. So again, in case these aren't familiar, I'll go through one quickly. So here we have a measure in twin 1, so it might be anxiety, for example, and we look at 3 variance components: additive genetics, common or shared environment. These are the 2 that I explained on the previous slide. And then non-shared environment, which is that captures those influences not shared between the twins that don't make them similar. So, for example, having different experiences, a measurement error goes into here as well. And when we think about the correlation between a pair of twins, there's 2 different routes to this correlation. First, they correlate because of genetic similarity, which is 1 for identical twins and 0.5 for non-identical twins. And then they can also correlate because of their shared environment, which by definition is 1 for both types of pair. And then the non-shared environment is specific to each individual, and it makes them different from one another. So one of the studies that I'm going to draw on quite a bit in my talk today is the Twins Early Development Study, and this was initiated in the early 90s by my long-term colleague, Robert Plomin. We've had 30 years of consecutive MRC funding, and in the most recent programme grant, I took over from Robert as the director. And we have numerous waves of assessment at these various age points, and the most recent of which is at age 26. Our next phase of grant writing is just beginning, and we'll be looking at mental health in the twins in their 30s. So this is a shout out to anybody who'd like to give me ideas for things they think it would be good for us to include in that wave of assessment. There were 13,000 families originally in the study, of whom around 10,000 are still actively involved. And in this age 26 assessment, around 9,000 twins took part. And there's a really nice description of that assessment written by Celestine Lockhart. It came out this year in JCPP Advances. We also have DNA on the twins, and a lot of measures have been repeatedly collected. So it's a really excellent study for longitudinal data. And you don't necessarily have to use the twin element of the data set, although it is beneficial for some kinds of analyses, but the longitudinal analyses don't necessarily have to exploit that. It's really easy. This is just a brief shout out about TEDS. It's really easy and also free to access our data. So I'd really encourage you to think about that if you're somebody interested in any of these kinds of phenotypes. We have 6 main domains. So I'm talking mostly about mental health today, which has increasingly become a focus. But we also have a lot of data on cognitive and educational achievement, measures of the home environment, more briefer measures but frequent of physical health and well-being, and a little bit on personality and motivation. And there's a really lovely data dictionary online that you can just access via our website. And there's a standard data sharing agreement you just complete of a data sharing request. And that gets attached to the data sharing agreement that your university signs, and it's a standard one provided by King's. So it's a very straightforward process, and we're keen to encourage people to use the data. So back to genetics. From twin studies, we know that anxiety and depression are both moderately heritable. If you look at them at a single time point, the heritability is typically around 30 to 40% of the variance, and the rest is typically non-shared environment. In terms of genome-wide association studies, these are beginning finally to take off. This is data from the Psychiatric Genomics Consortium Anxiety Group, which I'm one of the co-chairs for, and we are just writing up our current freeze with 61 genome-wide significant loci from a data set with around 118,000 cases in it. I'm not going to talk about GWAS as an analytical method. I'm just going to talk about how we use this data. We basically combine the more informative data in the data set and combine that into genome-wide polygenic scores. And actually, I should have said before mentioning that each of these individual dots, for those of you not familiar with these plots, is a single genetic variant. These are the chromosomes lined up along the bottom, and this is the level of significance, so the strength of association between that variant and your outcome, which in this case is lifetime anxiety disorder. So these green ones are the ones above the genome-wide significant bar of 5 × 10⁻⁸, which is effectively a Bonferroni correction for having done 1 million tests, which is a standard way of testing for statistical significance in these genome-wide studies. But as I say, we're going to look at those only in terms of combined genome-wide polygenic scores that combine the information at the upper end, not just the genome-wide significant ones, but some of the ones a bit below that as well. One thing to mention is that these genome-wide studies are really taking off now, but they need huge sample sizes. So this is a plot from a paper a couple of years ago, 4 years ago now, in fact, and along the x-axis is the number of cases in the analysis, 2,000, 20,000, up to 200,000. And then this is the number of independent loci identified. And you can see that we sort of grumbled along the bottom for quite some time, and then it's just begun taking off. So this anxiety paper I just mentioned is similar to the MDD Naomi Ray paper, for those of you familiar with it, that came out maybe 2 or 3 years ago. And there was then a subsequent larger paper also using UK Biobank data. And then now there's one in preparation at the moment with over 500,000 cases and over 570 loci. So it actually goes off the size of this plot. But that's just to sort of keep in mind the scale of data sets that we need to gain really useful genetic information, which has some implications for some of what I'll be talking about later. Okay, so on to the development of emotional problems now. And one of the models that we use most commonly in the twin design to test influences on development of traits over time is the Cholesky decomposition. And I've given an example here with 3 time points. So you've got the same A, C, and E, additive genetics, common or shared environment, and non-shared environment that influence, say, anxiety at time 1. But those same influences can also have an impact at time 2 and at time 3. So these are sort of longitudinal paths, basically. And then there's a new set of influences that comes in at time 2 that can also longitudinally influence time 3. And then there's always a time-specific set at the end for whichever is your final data point. And so influences from this shared one that impact all 3 are your sort of core stability measures. These are showing some stability, but some innovation compared to the earlier time point because they just come in afresh at time 2. And then this is a final wave of genetic innovation. So here I'm summarizing some data from 5 different scales that we looked at in 3 time points in a twin study of adolescents and early adults, age 15, 17 and 20. We've got generalized anxiety, separation anxiety, social anxiety, panic and depression. These were all self-reported scales. And the black each time is A1, so the first A term that starts off having an impact at age 15. And you can see it continues to have an impact at age 17 and 20 for all of the variables, particularly so for depression. There are also some new genetic influences coming in. This middle grey is A2 coming in at age 17 and showing a little bit of stability in some instances. And then there's a bit of genetic innovation at age 20 as well. But there's definitely more stability than there is innovation overall. And this is in contrast to the environment, where we see that the environmental factors at age 15 have barely any impact at age 17 or 20. And this applies to all of these different traits. And similarly, the age 17 seems to be pretty much time specific. So we've learned from this that the genetic influences on these sorts of emotional problems is largely stable and more influential on stability than on change, and the environment is more influential for change than for stability. So then taking that a step further, we've done some analyses of which this is the most recent, where we've extracted a latent stable measure. So this is generalized anxiety stability in the TEDS twins at age 21 through to age 26. And then these are 4 ways of assessment done during the pandemic. This is work being undertaken at the moment by Julia Funk with Geneviève Monod-Viancourt, who are both visiting the lab at present as PhD students and postdoc fellows. And what we find is that when you extract this stability, the heritability nearly doubles. So it's now 60% of the variance rather than the 30% that you typically see at a single time point. So this is very useful information when you're thinking about how to increase power to detect genetic effects. And in fact, because this finding has been in the literature for a little while, this is something I looked at with a prior PhD student, Rosa Cheeseman, where we looked at measures of anxiety and depression at different time points and different measures and parent and self ratings. And these are the heritability estimates for what's called the SNP heritability. So this is how much of the variance in these symptoms is accounted for by all the genetic variants, which are called SNPs, from the genome-wide array. And none of these is significant when you look at a single one on its own. But when you combine them together using either confirmatory factor analysis approach, similar to the prior slide, or item theory response scores, you then do get a quite substantial and significant role of common genetic variants, these SNPs, on emotional symptoms in young people that's approaching 18% of the variance. So then this is another way of thinking about longitudinal data and polygenic scores. This is work by Alex Kwong. And so he was building trajectories of depression symptoms using ALSPAC dataset, and then showing that the depression polygenic score and the major depressive disorder polygenic score, anxiety, neuroticism, schizophrenia polygenic scores, showing how they how people who were 1 standard deviation above the mean on that score versus 1 standard deviation below, which is the 2 kind of outer lines, how their trajectories differed. So you can see, for example, for the depression PRS, that those in the top +1 standard deviation of the mean for this polygenic score, they end up on this quite a bit higher trajectory, quite a bit higher than you see in the average group, and the group whose scores are at least 1 standard deviation below the mean are correspondingly lower, with very clear confidence intervals around them. So this is very useful to help us think about how these polygenic scores might be impacting on these traits over this developmental period, particularly given that these polygenic scores have all been created from adult samples. And this is work that Alex is building on with Ellen Thompson, and they've had a couple of phases of Wellcome Trust data prizes and have built this really lovely tool called Tidal, which allows you to build these kinds of trajectories, and it also allows individuals who don't come from a research background or don't have training in these kinds of models to do it in a very simple and easy way. So if that's something that might interest you, then do give a shout out to Alex. I'm sure he'd be happy to hear from you. So then this is data that probably some of you will recognise. This is also ASPAC data, but it comes from your centre. It's a paper by Weavers et al. That came out in The Lancet a couple of years ago. So this is another set of trajectories, again in depression, in ASPAC, and 4 different trajectory classes to try and tease out the kind of heterogeneity that you see. So overall, you see that pattern of increasing depression over time, but we know that that doesn't apply consistently to everybody. And so that what they found was a high group here, with that being about 7% of the sample, a couple of groups here in the middle that had quite changing trajectories, and then a stable low group here. And this is a relatively classic set of findings to see over this kind of developmental period for a trajectory analysis. And they found that childhood poverty and social adversity and child autistic traits were associated with being in the higher risk depression group. And in terms of outcomes, this higher group had higher levels of suicidal self-harm, poor functional impairment, mental health difficulties, and were more likely to be not in education, employment, or training. And thinking about polygenic scores, they saw that the major depressive disorder and ADHD polygenic scores distinguished these groups, and the distinction between these 2 groups was largely because of the major depressive disorder polygenic score. So we're beginning to pick up a sense that both polygenic scores and individual measures of experiences that children are having in their family can be used together to understand the likely trajectories of these young people. This is something we wanted to explore further in TEDS, and this is work by Elizabeth Palailegou, and she's a final year PhD student in the lab, and so she built symptom trajectories from age 4 all the way to 26. So that's a little bit different. These 2 ASPAC ones start towards the end of childhood, and they're on depression. This is from all the way back to age 4. So we wanted to use anxiety, as we know that begins very young, and it goes up to age 26. So we saw a somewhat similar set of classes with a high increasing class, a low increasing class, a moderate decreasing class, and then the same sort of low stable class. And this is the prevalence of each of those classes in TED. So it was just 3% in this top group, and 68% in the low stable group. And these are heritability estimates for being in one of these classes. The stable low group is the most highly heritable, which makes sense because we know that genes are most strongly involved in stability. And then the one that changes the least out of the other 3 is this relatively stable high group, and that has a heritability of 56%, with the 2 that change 52% and 49%. So we then had a look at outcomes of these different groups so that we could really fully understand who they were and what the risks were of having been in one of these trajectory groups. So here we're combining these 3 high groups against the low stable. So they're all compared against the low stable. And you can see that in general, in terms of outcomes, the decreasing group, there's not really a lot going on. They're not having too many problems. Whereas the high stable and the low increasing group are both showing difficulties in a wide variety of areas across different kinds of mental health concerns, functional impairment, experiences of negative life events, peer problems. And mostly this high stable and the increasing group don't differ from one another, apart from here where we see that in terms of current anxiety, the higher group has the highest scores for anxiety of all. And then these are some other kinds of dichotomous outcomes. So whether they've sought help for mental health, whether they've got a university degree or higher, this same NEAT outcome, not in education, employment or training, receiving benefits, being unemployed, suicidal thoughts and self-harm, alcohol dependence and cannabis, high addiction risk. And you can see that again, across the majority of these, we are seeing poorer outcomes on this wide array of different kind of variables for those in these 2 groups. And just to put that in a slightly different way, if we compare these high groups, the two high groups, to the decreasing and low groups, across sort of 6 main areas of outcome, we see that in terms of other mental health diagnoses, suicidal thoughts, and not being in education, employment, or training, they're at 4 to 5 times the risk of those in the low groups. We also find that they're 3 to 4 times at risk of uptake of benefits, and they're 2 to 3 times as likely to be experiencing impairment, for example, with their education or their work, and to be experiencing peer difficulties such as social isolation. So this is a very concerning group of young people that we want to better understand. So we then looked to factors that might predict membership of these groups. And because our groups started at age 4, we only looked at data in TEDS that was captured up to and including the age of 4 as a starting point. So above, in the upper half of this figure, the bit I'm showing you at the moment, are non-genetic factors. And these are ones that were significant in their univariable models, and we then combined them into a multivariable model. And in that model, you can see that there's still a significant effect of postnatal depression, of household chaos, and a really large effect of female sex, as we would expect. In terms of polygenic risk, we see a very large effect for the only well-powered polygenic score for internalizing symptoms, which is from a meta-analysis. And we also see a role for the ADHD PRS, a little bit similar to what we saw earlier from the Cardiff results with depression in the Ausbach sample. So this is contrasting the increasing group against the low group. I'm not going to show you all the pairwise contrasts because it would take longer than I have to go through them all, but happy to share details about more of them if there's specific contrasts you want to know about. So this is the other contrast I'm going to show, which is this high and increasing group versus the decreasing group. And here, really, the only variable that's not a genetic variable that survives in the multivariate model is female sex. And in terms of the genetic factors, it's really the childhood internalizing polygenic score that's a player. And the effects are basically additive in that what we're seeing is higher levels of an influence of sex and child internalizing score as we move from the bottom stable group up through the 2 changing groups to the highest group. So the highest polygenic score is seen in these high stable children. So then we really wanted to use this information together in a slightly different analytical framework where we're doing prediction analyses, kind of like an AI kind of approach. And so what we put into these was our polygenic scores and then everything that we had in the data set from before age 4 to do with their home and school and social environment, and to do with their traits and their characteristics and those of their parents that we had at that time. And that we find, depending on the particular function used, we get an accuracy of around 65 to 80%, depending, as I say, on the details of the analysis. So it's actually really quite encouraging that even with information that you would have age 4, you can have reasonable chance of identifying which of these groups young people might be most likely to fall into, as that would then enable some targeted interventions. So just one other kind of developmental outcome that we're really interested in the group, which is retrospectively reported trauma, which is very strongly associated with mental health problems, and in particular with anxiety and depression in children and adolescents and young adults. So this is work by Alicia Peel when she was doing her PhD in the group, and it's the same kind of set of findings. It's all the ones that were significant in the univariable model, and then we've put them together into a multivariable model. Again, the upper half of the model is the non-genetic factors, and here we've used information about environmental adversity measured by either the parent report or by self-report at these different life stages. And what we see is that the ones that remain significant in the multivariable model are childhood parent-reported environmental adversity at ages 7 to 10. So that's variables such as negative parenting, punitive parenting, and chaos in the home, these sorts of variables. And then adolescent self-report from age 12 to 16. So similar variables and also life events as well at that age. And then she looked at an array of polygenic scores and saw that the autism spectrum disorder polygenic score and the post-traumatic stress disorder polygenic score were both significant, remained significant in the multivariable model. So again, we're getting a similar kind of pattern that both the genetic risks and the non-genetic risks are making independent contributions to understanding these important outcomes. So a summary of what I've said about factors associated with emotional development. Postnatal depression can be important, as can female sex, as can various measures of social adversity, such as household chaos or parenting or social adversity, broadly speaking. And then thus far, we have evidence that the adult anxiety and depression polygenic scores are relevant. The ADHD polygenic score seems to be relevant, and the childhood internalizing polygenic score seems to be particularly relevant, at least for the anxiety trajectories. So now I'm going to move along to psychological treatment outcomes. And a lot of the literature here relates to adults, and in the adult literature, there's some evidence also in the child literature. For understanding psychological treatment outcomes, there are kind of 4 predictors that have been kicking around in the field for quite some time. So those with more severe levels of symptoms, those with a more chronic profile, those with comorbidities, for example, depression in the context of anxiety, and those experiencing social adversity all seem to do less well from psychological treatments. And this is something that we want to try and build on in a similar way to the work I was just showing you that we're doing on development. So this is one of the early studies from the team looking at this, and this was an international collaboration with Jenny Hudson and her team at Macquarie in Australia, Kathy Creswell, who at the time was in Reading, and a series of other child anxiety psychological treatment groups around the world, who all agreed to provide their treatment outcome data and collect DNA from the children so that we could look at genetic predictors of treatment outcomes. I'll come back to that a bit later, but this was a paper that Jenny led on that was looking at trajectories of outcome from baseline across post-treatment 3-month, 6-month and 12-month follow-up. And she's done separate trajectories for different anxiety disorder groupings. And these are children aged 8 to 18 years, and the majority of them are between 8 and 12 years. And as you can see in this figure, the children with social anxiety disorder saw the lowest level of clinical improvement. And also those with comorbid depression and comorbid conduct problems saw poorer levels of improvement. We looked at the impact of parental psychopathology, and that was sort of nominally significant. So it needs to be looked at again and potentially in larger data sets. So then going on to the adult literature, we have shifted our attention to using what used to be called IAPT, is now called the Talking Therapy Service. And this is work being led by Meg Skelton in the group. And this is one of her publications using data from the South London and Maudsley NHS Trust, which is the trust we're aligned to at the Institute of Psychology, Psychiatry and Neuroscience. And these are similar trajectories to the ones I showed you earlier. So here we have a group, it's the anxiety figure that I've got here, but they're actually very similar for depression, and in a different data set she's done them for impairment as well, and they all look pretty similar. So you have this high stable group, so effectively non-responders. And then we have 3 groups of responders who all end up in a quite similar place, but they go about it at a very different pace. So there's one group who start relatively low anyway and gradually seem to improve. Then there's a steep improvement group, so a group who really rapidly improve. And then there's a group who we're really interested in, who improve but quite slowly. And so there's other work going on in the group with Noor Kanzo and Ewan Carr, where we're going to be looking at how many sessions we need to have collected data on to be able to give a reasonable estimate of whether this person is in one of the improver groups and should stick with the treatment, or whether they're in fact in this non-improver group and a different approach should be considered. But in this paper that Meg did, we were looking at predictors of these group memberships because we thought that was a useful thing to explore better. And so on the next figure, we're looking at the contrast between these 3 groups, which are the 3 improving groups, against this high group, the non-improver group. And so this is a plot of Effect sizes anything less than 1 means that they are less likely to be in the improvement group. And these are the various factors we looked at. So comorbid symptoms and functional impairment, whether they're prescribed medication, whether they're unemployed, whether they have a disability, and whether they identify as Black, Asian, mixed or other in the data provided at the start of treatment. And you can see quite clearly that most of these factors are associated with one, if not all 3 of the groups that do well in a negative direction. In other words, they are less likely to be in one of those improvement groups if they carry one of these clinical features. And presumably, the more they carry, the more likely it is, and that's ongoing work in the group. So just to summarize, the factors associated with poor treatment outcome are comorbidity, taking medication in the adult population, unemployment, being out of work, having a disability, and being from minoritized ethnic or racial backgrounds. In terms of polygenic scores, the literature is very scarce, and we can't really say anything with much certainty at the moment. But using the study that I mentioned earlier, we created a polygenic score for differential susceptibility or responsivity to the environment. So this is the idea that some people respond and flourish if they're in good environments, but if they're in a poor environment, they'd really struggle, whereas other people are just fine regardless of the environment. So kind of contrast you might see between an orchid, which is beautiful if looked after well, but does need care and attention and support, versus a dandelion, which I can't tell you how many pictures there are of dandelions popping up through tarmac and pavements. So they're clearly very hardy. So we found some preliminary evidence that a polygenic score associated with this sort of responsivity to the environment was also associated with doing well in CBT for child anxiety if you had a kind of one-to-one form of the treatment, but not doing so well if the treatment was delivered in a more dilute form via a group or through parent delivery. And then Christian Rook's group in Sweden identified that the autism spectrum disorder polygenic score was associated with worse outcomes from internet-based CBT for depression. But these are really the only findings in the literature at the moment, and the main issue is power. That was the reason I showed you that slide at the beginning with the sample sizes. Sample size for both of these studies is about 1,000 to 1,500, that kind of size. And you saw that we're just beginning to get useful information when we have sort of tens of thousands of cases in our genome-wide studies. So although use of polygenic score doesn't require as much power as doing a genome-wide study, it is still a method that requires very big sample sizes. So we kind of decided we need to think again and not try and use these sort of individually collected clinical trials as our approach. And that's really why we moved into using the IAP service data, the talking therapy service data. But that doesn't come with DNA. So I'm going to tell you about another relatively new study that we've been running for the past 5 years. We just celebrated our 5th birthday, called the Genetic Links to Anxiety and Depression Study. The goal is to recruit 40,000 individuals who provide permission for us to recontact them and to link their medical records, including their talking therapies and IAPT records. They provide a lot of detailed lifetime history through a sort of online portal at the start. And this is an NIHR mental health bioresource, and it's funded as part of the overall NIHR bioresource. And we kicked this off on international news, on ITV News, national news, in September 2018, and we had support from several very relevant national bodies such as the Royal College of Psychiatrists, Mind, British Psychological Society, and they were really instrumental and essential to us getting the messaging right and making a success of this launch. And various individuals with lived experience and also people in the public eye who were interested in raising the profile of mental health helped us as well. So this is an example of some of the sort of social media that we had going on at the time. So this is an update on where we've got to with GLAB participants in November 2023. We now have over 34,000 participants who have completed everything. So they've provided consent, they finished their survey, and we've received their saliva kits. And so their DNA is now being genotyped by the NHR. And you can see from this map, I'm afraid is a bit older. We were hoping to get one updated for today, but somebody was having trouble with the script and a piece of our code not working anymore. So that's a little bit of an old one, but you can see that we've gone across all of the devolved nations. We have a lead in Wales, Northern Ireland and Scotland, as well as ourselves being based at King's in London. So many thanks to any of you. There probably are several of you on the call who have helped us with this, because it's been a really massive effort. And so the purpose of GLAD is not just to do analyses, but primarily to support recruitment into other studies. And recruitment can be undertaken using the self-report data that participants are provided, their DNA and all their medical records. And we're part of the Longitudinal linkage collaboration. And so we have access to the medical records of the GLAD participants. And so this can support all kinds of different studies. So there have been treatment studies, psychological treatment studies, medication studies, imaging studies, all sorts of things. Yeah, it's been really exciting seeing that happen and facilitating so much research into anxiety and depression. And the age range of that study is 16 plus. We don't have many that are 16 to 17, especially not men, but we have in general quite a young adult sample. So the majority of the participants are in their 20s and 30s, although we do have some right up into their 80s and 90s, which is fabulous. Okay, so I will wrap up there by saying, where could this all lead? And I guess my goal, and where I was hoping we might eventually get to when I first started working in prediction of treatment outcomes, which was coming on for 20 years ago, was the idea that if you had a young person walk into a clinician's office or a teacher's office or a GP's office, and you want to know how best to address their concerns, are they somebody that's going to benefit from a school-based intervention or a group-based intervention with a mental health professional, or perhaps they need one-to-one, perhaps even something else. And we just don't know very much about who will benefit from which of these approaches. So our goal is to learn how the combination of clinical information, demographic information, and genetic information together can improve our understanding of what the best match is for each individual, so that we can get them back onto a healthy pathway as quickly as possible. So massive thank you to all the wonderful people that I work with. I think I've mentioned most of them along the way, but in case I didn't specifically mention Jerome, he and I run GLAD together, and he's been a long-term collaborator supporting all the genomics work that we do in the group. And Ewan has supported all of the prediction modelling analyses that I presented. So I think I will stop there. Oh, actually, I have one more slide. If you're interested, we write a blog, our lab, and it's a mixture of pieces about our science and pieces about our lives as scientists. So there's a very nice one just came out a week or so ago, which is V in our latest A to Z. It's our second A to Z series, and it's V for Viva, with some thoughts about how a couple of recently completed PhD students prepared for their Viva, how they found their Viva, what they learned from it, and they want to share with others who've not yet done theirs. Oh, that's just gone blank on me. Am I still there? So I will stop there, and very happy to take any questions.
Yeah, thank you. That was lovely. And you stuck with 45 minutes, so that's great. I was just looking, for a question that came up from Anita. It says, a common reason for treatment-resistant psychological or medication is a misdiagnosis or misdiagnosis. For example, missing PTSD or a neurodevelopmental disorder in young people or adults with depression or anxiety. Isn't that a challenge with IAP data and for GLAD, unless you look at all psychopathology? Yeah,
absolutely. So I think I'm not a clinician, but my understanding is that in IAP data, individuals with more severe mental health conditions are referred to other pathways, but I'm sure some people with milder presentations, for example, of autism or ADHD, or particularly females with less well-known presentations, do slip through, and I'm sure that would have an impact on outcomes. Absolutely. I don't think there's consistent data being collected on that, so it's not something we can look at in the Talking Therapies data sets. In GLAD, we have very detailed measures of lifetime experiences of anxiety and depression, but we have brief measures of pretty much every aspect of psychopathology you can imagine, including all of those conditions. So that is something that we could look at when when we connect our data with the talking therapies records or look at other measures of treatment outcome.
Yeah, thank you. So the next question is, what are the main challenges the field team faces when conducting longitudinal research and collecting the data?
Keeping them in. I think that's the main challenge. It's really hard keeping your participants engaged. I mean, I think in TEDS we have an advantage that they're twins, and twins are typically more committed to research about being a twin than singletons are, because being a twin is such a key part of who they are because of it being unusual and special. So that does help, but we still lose people the same as everybody else. And it's something that we have to put significant effort into addressing. And we have some of that happening sort of constantly all the time. And then we have periods of really strong activity in that area. So it's a mixture of those two approaches, really. I would say another thing is that if you want to do a really good job of running a longitudinal study, you want to try and figure out when you're hiring who are people that might want to stick around for a long time, because as far as I'm concerned, TEDS is the team, and there's 3 people in particular in the team who've been in the team for around 20 years and who I feel like I've worked with pretty much my whole career. And they are TEDs, really. So I think establishing really good relationships with the people, running the study, running the data, looking after the participants, that makes a huge difference. And our twins all know our project manager really well. They know who she is and they like hearing from her, because she's been around for such a long time. So I think that's an important part of keeping it going as well. And then I would say the other challenge is funding. Enough said.
Yeah. So there's a follow-up comment from the first question. So from Anita, so we found in our study of adults with recurrent depression, those with high persistent depression had a higher than expected rate of ADHD, for example, which is not picked up by hospitals or GPs. So yeah, sorry, it was just a
interesting. So I mean, that suggests that, I think Elizabeth is on the call actually, that suggests that in our high group, we, as I explained, we very much looked at features of these children before age 4 so that we could be thinking about predictors. But it might be interesting for us to have a look at other features of psychopathology in, for example, adolescents, and see. In terms of outcomes, we did definitely have more mental health diagnoses in those higher groups. Whether we've actually looked at whether they were picked up by a GP or hospital or not, I'm not sure we've looked at it in that level of detail, but it's certainly something we could explore. Be happy to chat about that more.
Yeah, thank you. So the next question is about, it says, the treatment response predictors you highlight, for example, comorbidities, are not specific prognostic predictors that would predict poor outcomes regardless of treatment.
Yeah.
Have you looked at what might modify the treatment? That is, what predicts whether a particular treatment is suitable for a particular person?
Yeah, it's such a good question. We're trying to. That's the goal. It's just, it's really difficult. And I think one of the reasons it's difficult is because it's such a complex phenotype. So thinking about anxiety and depression at a single time point is complex enough, but thinking about treatment response is effectively change in those symptoms in the context of something else happening. Which adds a whole nother layer of complexity, and your outcome is sort of automatically a different score, which has more error in it as well. So I think there are lots of methodological / conceptual reasons why it's challenging. And I think these sort of prediction models taking an AI kind of approach are the way forward. And that's what we're building towards in the team. But the IAP data set itself doesn't necessarily have enough information in it. So that's why we're wanting to connect those records to GLAD, and we're also in the process of connecting them to TEDS as well, so that we can try and bring in some information that wouldn't typically be clinically collected, including genetic information, but not only, and see if that helps us get a bit closer. But yeah, you're right. The information is all very kind of common sense, and you could have guessed it really at the moment.
Yeah, I was in terms of the part you're talking about, talking therapy services. it was very interesting to see that there were like 3 groups were improving that had similar outcomes, and like at the end of the study period. And I was wondering, you might have mentioned it, but I was wondering if you were able to look at those who like dropped out at some point during the treatment period. I know you said that it was data given to you by other people that were undertaking those services, those treatments. So yeah, I was wondering if you could, it was possible to see those who did not complete the treatment and whether they differed from the others.
Yeah, so we did have information on who dropped out. I mean, you could infer it from what was no longer there in the data set. And we didn't look exactly at what you've just asked, but it is one of the things that we're looking at in the next project. We're thinking of it as a kind of outcome in its own right. So just looking at prediction of dropout. So it's on the list, working our way down the list.
So I was also wondering about like an earlier study you mentioned about the trajectories of anxiety symptoms. And you did mention also a study that's been done by Briony Weavers, which was on depression trajectories. And I know that study was using self-report. That's why I started a bit later during childhood, like around 10. But I was wondering, because the study you mentioned on anxiety started from age 4. So I'm guessing that it will be like a lot of parent reports. And I'm wondering whether you think, how do you think that might change the fact that so it's an individual might be grouped as high persistent or an adolescent increasing based on like those early time points?
Yeah. So what we decided to do for the anxiety trajectories was to use both parent and self-report. And at the ages where we only had one, we just used that one. So by the time they're age 26, it's all self-report, and at age 4 and 7, it's all parent report. And in between times, it's mostly both. But also some people would only have had one. So if we only had one, we went with what we had. As I recall, I haven't looked at it for a little while. Elizabeth, do stick something in the questions if I say the wrong thing. As I recall, we didn't see huge differences when we just reduced it to working with the parent-reported data only and the self-reported data only. Obviously, they sort of had this overlap patch, but there were sort of areas at either end of the age range where they didn't. So the trajectories themselves didn't look particularly different. I don't think we actually looked to see whether individuals ended up allocated to the same trajectory using each approach, as it were. Yeah, we were reasonably comfortable with our approach. The main thing we know about parents and internalizing symptoms is that they're not great at telling the difference between anxiety and depression, for example, but they're reasonably good at telling that there's something going on in that area. You don't see as high correlations between parent and self-report as you would between, say, a self-report measure of anxiety and a self-report measure of depression, but they're in the kind of 0.4 to 0.5 range, so you know, not too bad. I think if you were doing it on other traits, I was discussing with some colleagues about doing some trajectories in conduct problems, and they were making the point that for those it would really be essential to use parent reports, because the young people would typically who have a tendency to be a bit naughty tend not to be all that truthful as well. So I think it kind of depends on what your phenotype is, and I think it's not perfect doing this, but because we wanted to cover that full age range, there isn't really another option, so it's kind of a pragmatic approach as well.
Yeah, Elizabeth just supported what you said. Said trajectories look the same.
Thank you.
And there's another question.
Oh, I can see it, yes.
Yeah.
Okay, why was the anxiety polygenic score not as good a predictor as the internal polygenic score, the anxiety trajectory? So the anxiety polygenic score is an adult sample. So, I mean, it's actually a better powered sample, at least it's from a larger GWAS, but it's from an adult sample. So my guess is it's a developmental feature. So if you think back to the slides I showed of Monica Vajkut's work on stability and change, and how most of the stability was genetic, there was still some genetic innovation. And I think if you're going from, you know, child and adolescent trajectories right into adulthood, you're covering a huge set of developmental periods. So I suspect it's that, but we don't really know yet. Once we've got more polygenic scores from both adult and younger samples, it'll become a bit clearer, I think. But yeah, we were pretty struck by that. And that's our explanation at the moment.
Yeah.
Well, if anyone does have any further questions, my email is very easy. It's just thalia.eley@kcl.ac.uk. So yeah, be happy to hear from you about any of the work, or about using TEDS data, or things you think we should collect in TEDS as they turn into their 30s. Yeah, we want to hear any more about any of the analyses.
Oh, there's the final question. So how is any of the genetics data combined with imaging data?
We haven't worked with imaging data at all. There are big consortiums that are combining imaging data with genetics data, so we're aware of those, but we don't have any imaging data in TEDS. I think some collaborators are planning to get some imaging data in some of the GLAD participants. There may even be a study underway now doing that, but it's not data we're working with.
Okay, thank you. And I think we're right on time to wrap up. And again, thank you so much for that, Ilya, and thank you everyone who joined in today. And as I said, have a look at the chat in case you want to find the links for the YouTube channel or the mailing list. And yeah, thank you everyone for joining in.